Hello, welcome to our Genes and Consequences presentation. Today we're going to be focusing on a specific patient and their issues that they've been having, and then what we found when we looked at their actual sequence for this gene to determine which kind of mutation they had that caused their problems. So to begin, we learned that our patient is a 36-year-old male, and he visits the doctor for arthritis-like joint pain, chronic fatigue, and abdominal pain. And his doctor's recommendation for how to alleviate these symptoms was to regularly donate his own blood. So, a gene overview after we looked at his sequence, when we used a research database, we found that it was the homeostatic iron regulator, or HFE gene, which codes for the HFE protein. And the location of this gene is on the sixth chromosome, and it's on the short arm of the chromosome. And the specific gene locus is 22.2. And here we have a 3D image of this protein. And the function is to regulate iron absorption in the gut via transferrin and the transferrin receptor by binding to iron in the bloodstream and transporting it into cells. So the mutation description, after analyzing the sequence, we found that a cysteine residue was changed to a tyrosine residue, which is known as a missense mutation when you switch out a specific amino acid residue. And this change in amino acids stops the protein from reaching the cell surface, and it causes the cells to take up too much iron. And the spot where this mutation took place was the 282nd amino acid in his sequence. And this mutation is recessive, meaning that you have to get the mutation from both parents in order to develop this disease. And here we have an image where we have a normal HFE gene and we have the cysteine residue, and then we can see in the mutation where the cysteine was turned into tyrosine.
Okay, so the main disease is hereditary hemochromatosis. It absorbs too much iron from the diet, leading to potentially serious conditions such as arthritis, liver disease, diabetes, cancer, and more. The diagnosis is found through blood tests that check for the gene mutations. The prognosis is that most people have a normal life expectancy if symptoms are treated. And a treatment is phlebotomy, removal of blood to lower iron levels in the body. And here we have an image showing how the hemochromatosis works. Other consequences of mutations in this gene are porphyria. The disease is abnormalities with heme production, buildup of toxic levels of porphins, iron-containing molecules in the liver and other organs. The diagnosis is found through blood and urine tests to screen for porphins, genetic testing. The prognosis is most cases are mild but can be life-threatening if severe. There is no cure. Treatments are aimed at managing symptoms. The treatment is injections of hemin, form of heme, to stop the body from trying to produce more hemoglobin. Other consequences of mutations in this gene: X-linked sideroblastic anemia. The disease in it is an HFE mutation exacerbates a mutation in the ALAS2 gene and leads to a buildup of iron because the body is not making enough red blood cells, and it causes an increase in heart rate, dizziness, and enlarged liver and spleen. The diagnosis is found through blood tests to check for complete blood count. The prognosis is that symptoms range from mild to severe, only life-threatening when untreated for a long period of time. And the treatment is phlebotomy and vitamin B6.
So for the sole determinants of the gene, this gene is indeed the sole determinant of hereditary hemochromatosis. It is also an inherited disorder. However, very few people actually suffer consequences from the symptoms. Would CRISPR work? The targeted approach is that the HFE protein is primarily found at the surface of liver and intestinal cells, and it could be possible to only target these organs. Unintended effects of this is the gene is specific to iron absorption. If corrected, it could possibly cause the opposite effect with too little iron absorption, but it is unknown whether the mutation should be completely knocked out or just tuned down. Other HFE gene mutations that cause hemochromatosis. There are more than 100 mutations in the HFE gene that cause hemochromatosis. And another mutation replaces is with HIS with ASP at position 63. And this is known to alter the 3D shape of the protein. And that is it. Thank you.
Thank you again for watching our presentation about how a gene sequence relates to a disease and whether CRISPR would be able to help with that disease.